Enzymes. Okay. So what do you mean by enzymes? See, enzymes are called biological catalysts, we know, right? Enzymes are called biological catalyst. Okay, so what is the role of a catalyst? In chemistry, we know what is a catalyst, right? What is a catalyst? Catalyst can increase the rate of the reaction. There is a positive and negative catalyst. Some catalysts can increase, some of the catalysts can decrease. Okay, there are two types of catalysts that you know, positive and negative. The catalyst which can increase the rate of reaction is positive catalyst, decrease is called negative catalyst. Anyway, and the catalyst can enhance the rate of the reaction or they can influence the rate of the reaction. Influence means they can increase or decrease like that. Okay, so so normally we discussed about different catalysts. No, the transition metals are behaving as catalyst in different process, right? In organic, we studied about nickel catalyst, palladium catalyst, right? Then in Haber process, iron is catalyst. In contact process, vanadium pentoxide is catalyst. We discussed about catalyst. They are not biological catalysts. They are not working on our body. They are working in some chemicals, right? So it means outside our body they are working. Then biological catalyst means they are working inside our body in a biological system. They are working in biological system. Other catalysts means they are not working on our body. They are working on some chemical reactions outside the body. Platinum or palladium metal is not there in the body, human body, right? Yes. So biological catalyst means they are working inside the living body. Okay, so they are biological catalysts that initiate various chemical reactions in living organism. The reactions takes place in body under mild conditions with various sequences of chemical steps involved. I told you. See how the enzymes are active in our body, how they have become active. The same thing as I discussed in case of denaturation of protein, right? So enzymes will be active in our body in certain conditions only, mainly under mild conditions. Okay, and there is a particular steps for the enzymatic action also. Enzymes have the nature of globular protein. I told you, we know what is globular and fibrous proteins, right? The coiled one. Enzymes are reaction specific and act as a particular reaction and a substrate. See, enzymes means all the enzymes can do every function. No. For each and every enzyme, there is a specific function. Means enzyme actions are specific in nature. Enzymatic actions are specific in nature. They are not like all the enzymes are using for all the reaction. No, they have different roles. Okay. Then enzymes are named after the compound or class of compounds they work on, because whatever the reaction they are catalyzing, based on that, then only we can give them name. Okay, one example. Maltose is hydrolyzing into glucose. See, maltose is hydrolyzing into glucose. We discussed about this reaction. You remember, disaccharide. Disaccharide is hydrolyzing into two monosaccharides. This reaction is hydrolyzed by maltase. Maltase is the enzyme. How the name is maltase? Because this enzyme is helping for the hydrolysis of maltose. That's why they are called maltase. That's I told you, the name of the enzymes are given based on their function. Whatever the function they are doing, based on that we have to give name. Oxoreductase enzymes simultaneously oxidize one substrate and reduce. Means understood. Means some of them will be doing some purpose, some function. Some of the other one will do other function. Whatever the functions they are doing, based on, we have to give them name. Okay. Then what is the mechanism of this enzyme action? Next one. See, a small quantity of enzymes is required for the reaction to proceed. See, normally in a particular reaction, the catalyst, we need more catalyst, because catalyst, how the catalyst is acting, not in biological I am telling, normally how the catalyst is reacting. They are providing surface for the reaction. right? They are providing surface to conduct the reaction. That's why they are increasing that's how they are increasing the rate of the reaction. Okay. Enzymes reduce the magnitude of activation energy similar to activity of chemical analysis. You remember this? You know this diagram? Suppose in a chemical reaction, this is the reactant, this is the product. This is the energy axis. Okay. In a particular reaction, reactants are converting into products. Okay, understood. Then what will happen? The reactants are converting into products like this. So the peak of this one, this is called activation energy of the reaction, EA. What is activation energy? For every reaction to happen, the reactant molecule have to cross some energy barrier, right? Suppose a reactant molecule is this side. When they convert into product, see, when they cross this barrier, then only they will convert into product. This barrier is called activation energy. Okay, so we know collision theory, right? In every chemical reaction, the reactant molecules should collide with each other. Collide means they have to hit together. Then only the chemical reactions will happen. The reactant molecule have to collide together. They have to hit together. Then only they will convert into product. Okay, but the thing is, when the reactant molecules are colliding together, all the collisions are not leading to the chemical reaction, we know. The colliding molecule should have some minimum energy. Then only that collision will causes the reaction to happen. Okay, so that energy should be equal to the activation energy of the reaction. In every chemical reaction, there is an activation energy barrier. In every reaction, it is there. Okay, so if the reactant molecules have no sufficient energy to cross this barrier, the reaction won't happen. Okay, so the reactant molecule should possess minimum energy. That energy is called activation energy of the reaction, because then only actually it is the energy of the reaction. If the reactant molecules are not crossing that barrier, they are not converting into product. That's why we are telling us all the collisions are not successive collisions. So you studied, right? Yes. So I'm not talking about that too much because all this you know. Okay, then see, then what this enzyme is doing? When the activation energy is increasing, the rate of the reaction will decreases. See, suppose in a high jump, the height is here. So suppose 10 students, they are crossing this height. Okay. Then we have 20 students. The 10 students only, they are able to cross this barrier. This is the height. Suppose I am decreasing the height. I am reducing it. So 5 more students are capable of crossing this one. Got it? So this is the activation energy of the reaction. Now from the 20 molecules, 10 molecules are able to cross this barrier. Suppose this energy barrier, this is activation energy, right? Suppose this activation energy is decreased. Then what will happen? More molecule will be eligible to cross this barrier. Same thing is happening in enzymatic action. Same thing is happening in catalyst reaction, right? Same thing is here. See, this is the normal activation energy of the reaction. Suppose the same reaction I am carrying out in presence of a catalyst like enzyme. So what will happen? This is the reactant, this is the product. So they will, this is the chemical reaction in presence. I will draw again. See, here I will draw. See, this is the activation energy of the reaction. Suppose I am using a catalyst, the reaction will be like this. What it mean? See, this is the activation energy. This is decreased. So this is the activation energy. I will draw a clear diagram. It will be better. Okay. See, a particular reaction, reactant. Suppose the reactant have high energy. This is suppose this is an exothermic reaction. Okay. So this is the chemical reaction. So this will be the activation energy. This one. Okay. Now, in presence of catalyst, the reaction is going like this. See, now what is the activation energy? This is the activation. So this is in presence of enzyme. This is absence of enzyme. Okay, means catalyst. Okay, see, so what the catalyst is doing? Catalyst is decreasing the activation energy for the reaction. They are helping for the reaction to happen so fastly. One example, general activation energy for an hydrolysis, acid hydrolysis is 6.22. In presence of enzymes, the activation energy for the acid hydrolysis will be 2.15. See, what a difference, 6.2 to 2.15. So in presence of a catalyst, the energy will be reduced. Got it? That's all.